Welcome to the last video in the unit on prokaryotic cell biology. In this video, we're going to talk about the variety of internal structures that are found in bacterial cells. It's important to recognize that just because prokaryotes do not have internal membrane-bound organelles, they still have a lot of internal structures that allow for organization of the cytoplasm. Let's go ahead and take a look. We're going to look at a variety of internal structures of bacteria. Some of these internal structures are found in all bacteria, and in fact, they're found in all prokaryotes. Those are the ones on the slide that are marked by an asterisk. The other structures are only found in certain species of bacteria and provide specialized features and functions to those species. We aren't going to talk about the cytoplasm today. Even though it is an internal structure, you should be relatively comfortable with what the cytoplasm is. Instead, we're going to begin with the nucleoid region. The nucleoid region is the area of the cell where the DNA is found. While this area is not surrounded by a membrane the way a eukaryotic nucleus is, it is a distinct structure in the cell and can be seen in TEM images because of the difference in density. The nucleoid region contains packaged up DNA, which is packaged by folding into large loops and held together by DNA binding proteins, like you can see in this image. These bacterial chromosomes are typically circular and somewhere between 500,000 to 10 million base pairs in length. Compare that to the human genome, which is 3 billion base pairs. It shows you just how much smaller bacterial genomes are compared to ours. There is one origin of replication in the genome that's physically attached to the cell membrane itself. This is important for DNA replication and cell division, which we'll talk about later in the semester. Now we're going to move on and look at the ribosome. Ribosomes should be pretty familiar to you guys from other classes when you've learned about translation. Ribosomes are found in all living cells. Their structure is composed of both protein and ribosomal RNA, which function to translate proteins from messenger RNA. The ribosome itself is made up of two subunits. The small one, called the 30S subunit, and the larger one, called the 50S subunit. Together, those subunits come together for the full ribosome, called the 70S complete ribosome. You should notice that the numbers are based on sedimentation rate, which was a measure of size, but it's important to note that the size of the two subunits individually does not add up to the sedimentation rate of the full ribosome. So that is why you have a 30S subunit and a 50S subunit, but when together, they're only a 70S ribosome. While the ribosome functions similarly across all domains of life, there are slight differences in its structure, with eukaryotic ribosomes having a complete size of 80S. This becomes important because differences between bacteria and eukaryotic cells can become targets for antibiotics because they will damage the bacteria but not harm our own cells. We'll talk about ribosomes more when we go over translation. The next internal structure we're going to take a look at are DNA plasmids. Plasmids are important structures to the bacteria that contain them. They are considered extrachromosomal pieces of DNA, which means they provide additional genes to the cell that are not part of the actual bacterial chromosome, where the cell's genome is located. These plasmids can be various sizes. Some only have a couple thousand nucleotides and a couple of genes, while others are bigger and contains hundreds of different genes. The key is that plasmid genes are not required for the cell to be alive, but instead can provide extra bonus features for the cell. For example, many genes for antibiotic resistance are found on plasmid DNA, which gives the cell an advantage in the presence of an antibiotic. Now we're going to take a look at inclusion bodies, specialized storage structures found in prokaryotic cytoplasms. Although prokaryotic cells do not have membrane-bound organelles, they do have various inclusion bodies that are often used to store different molecules or perform key functions. For example, these large droplets seen in these cells here with the clearings are lipid droplets, storing the lipid for future use. While these dark spots are storing glycogen, and these spots are sulfur granules. Other types of inclusion bodies include gas vesicles, which are seen here in D. Gas vesicles are interesting because they can allow the cell to control the amount of air that's inside it, which changes its buoyancy. These are often found in aquatic microbes because it allows them to control where in the water column they can be. And then in E, we see structures called magnetosomes, which are really neat. Magnetosomes allow the cell to orient on the Earth's magnetic pole, kind of like a compass. And so it allows them to control the direction that it's swimming in. The last type of inclusion bodies are called thylakoids, and those are involved in photosynthesis. If you've learned about photosynthesis in the past, that term might sound familiar to you, because these structures are also found in chloroplasts, and they're responsible for performing photosynthesis in both bacteria that have them and in plant cells. The reason they're found in both cyanobacteria and eukaryotic chloroplasts is that chloroplasts are believed to have originally been free-living cyanobacteria that were engulfed by a eukaryotic cell millions of years ago, and over time they have lost the properties that made them a free-living cell. This is called the endosymbiont theory, and it also explains the origin of mitochondria. The last internal structure we're going to take a look at are the endospores. Endospores are found in certain species of bacteria and allow them to survive in conditions where the environment becomes harsh and unable to be lived in. The final internal structure we will look at is found in only a handful of gram-positive bacterial species, but is very important to healthcare. Endospores are a protective structure that certain bacteria can form, which allows them to take on a dormant state. The endospore is metabolically inert, which means it has no metabolism or enzymatic activity. It's not living and growing. However, what it is doing is protecting the DNA from a harsh environment. Endospores are very resistant to both physical or chemical means. And so they don't get destroyed by things like heat or UV light. They're also very difficult to destroy with chemicals such as antibiotics or bleach. You can visualize endospores if you do an endospore stain. They will appear as small green circles, seen here in this image, inside the red vegetative cells. A vegetative cell is just another word for the metabolically active cell. Endospores are most relevant to healthcare because they are produced by the pathogen Clostridium difficile, better known as C. diff. This makes C. diff very easy to spread through a healthcare setting because often standard room cleaners will not destroy the endospores left behind by an infected patient. This is why C. diff patients will have special PPE precautions in a healthcare setting. The other pathogen of note that makes endospores is Bacillus anthracis, the causative agent of anthrax. It is anthrax spores that were distributed through the mail in the 2001 anthrax attacks. When an endospore-forming cell ends up in a harsh environment, such as an environment that lacks nutrients or is harsh because it's become acidic or some environmental change that could kill the cell, the vegetative cell undergoes a process called sporulation and begins to generate the endospore. First, the DNA must replicate. Then, a membrane is built around that new copy of the genome. That cell membrane will form around the genome and then become surrounded by additional membranes that continue to surround it and become packed with calcium and dipleconic acid. This forms the protective cortex that will be the innermost layer of the endospore. Next, a layer of protein is going to form around the cortex, forming the protein coat. Once the spore is finished, the vegetative cell will die, releasing the spore to the environment, where it just sits, not being destroyed by the harsh conditions, but also not growing. When the environment changes and becomes favorable again, this will signal inside the endospore to cause it to germinate and grow into a vegetative cell that can begin growth and replication. Spores can sit for hundreds of years in the environment before they successfully germinate into vegetative cells.